Hi, everybody. I'd like to welcome you to our presentation today on antibodies. My name is Emily Pinnock, and I am a board member of the Salt Lake Alumni Club Board at the University of Utah. I am super lucky to both be an alumnus of the University of Utah. I completed my bachelor's degree in health policy in 2017 here, but I'm also a current student at the U. I'm a third-year pharmacy student in the College of Pharmacy. And so a few months ago, when we were discussing as an alumni club board who we could invite to talk, a professor that's maybe related to pharmacy or research or something, I thought of Dr. Owen. So Dr. Shawn Owen is assistant professor at the College of Pharmacy, and he is an assistant professor of pharmaceutics and pharmaceutical chemistry, which sounds very fancy and impressive. He's also an alumnus of the U and has 3 degrees from the U, which is pretty cool. A degree in chemistry, Chinese, and a doctorate in pharmaceutics. His research really centers around antibodies, and so he is the perfect person to discuss this topic. And I can confirm that he is a very well-respected and engaging instructor and lecturer. One of the courses he teaches at the College of Pharmacy is on pharmacokinetics and pharmacodynamics, which sounds like a really daunting class, but I can confirm that it's a great course. So I will go ahead and pass it over to Dr. Owen.
Well, Emily, thank you very much for that introduction. I appreciate it. And just so everybody knows, Emily already got her A. She's out of the class, so there was no bribe needed here whatsoever. But really lucky to have people like Emily that are supporting science and bringing this to all of the alumni out there. I'm really excited to share a few slides with you today and some information. And so I'm going to open up a presentation. And so just as a little bit more of an introduction, I'm an assistant professor in pharmaceutics. I do hold adjunct assistant professor positions in other departments. And the real value of those is kind of getting in interactions with other students and other faculty across the campus. As Emily mentioned, I am an alumni myself, and then was recruited back to the University of Utah as an assistant professor a few years ago. And since then, a main focus on my lab has been on antibodies. But what we do with antibodies might not be typically what you think of with antibodies, but I think it's going to be important today. I want to spend a decent amount of time really kind of informing everybody what antibodies are. And then once we talk about that, what they are and how they're useful naturally for the body, talking about what we can do with them to create new therapeutics and diagnostics and how we use them in the lab. So antibodies. So first thing that I want everybody to know is that antibodies are not necessarily antibiotics. So don't want you to get these two things confused. But an antibody is this. This is a crystal structure of one type of an antibody. But that might be meaningless just looking at this fancy image on the screen. So a few facts about antibodies. A lot of times antibodies we hear are called immunoglobulins. And in fact, the IG that is used in the abbreviation of most different types of antibodies, that's where it comes from, is an immunoglobulin. This is a globular protein that's used in the immune system. These are large and usually Y-shaped molecules, as you can kind of see from this structure. In this image on the bottom left, what you can see is another representation of an antibody, but I think it's really important to look at it in context with other molecules. And you can tell if you come all the way down here, this is the size of aspirin. And a molecular weight of about 180. But with a monoclonal antibody, it's almost 100 times that size. So these are very, very large molecules that are made up of different components. You see it's about 5 times as big as EPO or erythropoietin, and maybe 30 times as big as insulin. So these are very large molecules that are developed by and produced by the body. And so I have to stop and be amazed for a second, because if the body is willing to put so much energy into creating such a large molecule, it's got to be important. So these are antibodies are the mediators of the immune system, and they're kind of their main function is to bind and block antigens. And I'll go into more detail about the difference between an antibody and an antigen in a minute. But the other main function is that antibodies signal other proteins and cells to help with their different mechanisms. The antigen itself, what that means is a molecule or a structure that an antibody binds. In fact, the name antigen comes from the abbreviation for antibody generator. So a protein or some other molecule that can when it's expressed or presented in the immune system, it can then generate an antibody. So an antigen is what an antibody binds to. A little bit more about the structure of antibodies, and this becomes important later on. So they have what we call heavy chains. So these are large polypeptide chains, and there are two heavy chains. These are the purple parts, and then there are two light chains that you can see in the light blue. They're also composed of sugars. So they have these really important saccharides, a sequence of sugars that are down in this core that help with some of the signaling that is required of antibodies. There's one part of the antibody. This is the part that actually binds to antigens. It's just this arm. So if you ever see somebody drawing an antibody with something bound in the middle here, it's wrong. And you can point that out to them from now on. So this is the part of the antibody that binds, and it's called the antigen binding domain. This whole piece is actually referred to as an antibody fragment, or the antigen binding fragment, or a Fab. And this other part is the portion that signals the immune system. And this is the crystallizable fraction, or the FC domain. So again, this is the part that binds. This is the part that really helps signals the immune system. But as I mentioned, this is just one structure of one type of antibody. There are actually a lot of different flavors of human antibodies. Now, just full disclosure, when we say flavors, I think this is a term that's rather loose. It doesn't mean that I actually know what these taste like. They are just different constructs. And looking at the different things that we have here, there's IgG. This is probably the most important one, especially for therapeutic antibodies, and it's the one that's most important for different functions. But what I want to point out is that all of these different constructs, IgD, so again, immunoglobulin type D, type A, M, and E, they all have this similar core structure where you have heavy chains and light chains, and these two pieces come together to have a binding domain. And then they have these polysaccharides that are very important in different functions. Now, I understand that not everybody probably wants to sit there and stare at a crystal structure as much as I do. So I'll use cartoon structures for the rest of the talk today. And so this is kind of a representation of these different pieces. And so what we have is IgD, G, E, and M, and A. And I'll talk about some of the different mechanisms that are really important. And these are just the basics. Sorry, this got cut off. So these are the natural antibody mechanisms. These are just the basics. There are some other extensions of these. But when we think of antibodies in general, it's most important in an infection. So you get an infection, some kind of a bacteria or a virus, and the body wants to deal with this foreign invader. So how does it clear this from the system? So one way is that it can actually have antibodies that will bind directly to this kind of a pathogen, this bacteria or whatever else it is, and a process called opsonization. This means it makes it, literally that translates into making it tasty. And so then once this becomes opsonized, these special immune cells like macrophages or neutrophils will then interact with the signaling portion of the antibody and eat whatever has been presented to them and destroy it. A second mechanism is called neutralization. And so what happens here is a lot of these molecules, like viruses, they have one part of the virus that's very important for infection. So this part of the virus might bind to something in the body and then hijack that to get internalized and infect the cells in our body. And so antibodies can be developed that will block that actual interaction and neutralize it, preventing any kind of infection from happening. Another more direct method of killing is what is known as complement activation. So in this system, antibodies first will bind to this target infection, this pathogen, and then recruit another set of molecules, these complement proteins, that will then actually cause pores to be formed. So they'll release additional molecules that will directly impact the infection itself, ripping holes into it and destroying it that way. A fourth mechanism is what's called antibody-dependent cell killing. In this mechanism, what you have is an infected cell that has different kind of antigens on the surface. Antibodies will bind to that cell, and through that, they will then signal, again, other large cells in the immune system, such as macrophages, neutrophils, and these will come and through this mediation will then launch an attack against the infected cell and then killing it that way. So all of these, you can see that antibodies are this really important central mediator in helping to fight different types of infections. Specifically, a little bit more detail, the IgD, what it does, it doesn't have a major role in the immune system, but what it does, it really is important in helping develop other antibodies. So it's kind of a mediator of this process. IgAs, these are really interesting, and you can see that I've drawn it here as kind of this back-to-back dimer of two IgAs. These are really important in secretory pathways. So in the gut, in other places where pathogens might be exposed. It really protects strongly against pathogens. What's interesting about IgA is I like to call it your mama's antibody, because this is the IgA that actually is presented in mother's milk before an infant has their own intact immune system. And these IgAs that are presented that way can actually be incorporated into the infants and help to fight off pathogens before they're able to do so on their own. Other molecules like the IgE, this is very important in the allergic reaction response. IgG, again, kind of, my opinion, the workhorse of all of the different antibodies. We have this really important in virus neutralization, in opsonization, and in a lot of other mechanisms. And its partner is this other called IgM. This has really similar functions to IgG, but usually is presented a little bit earlier in developing immunity. So I know that everybody is, obviously, we're all really, our day-to-day has been impacted quite a bit by a virus, particularly this virus, the SARS-CoV-2 and COVID-19. So I really want to talk a little bit about COVID and about how antibodies naturally arise against this infection, but also some other things that we can do to help develop them. So just as a little background, for those of you who don't know at this point, coronavirus structure, it turns out that coronaviruses are just a family of viruses, and other ones that we've heard in the past, in my lifetime, the first SARS, and then MERS that came out in 2012 and 2015, and then the more recent pandemic with SARS-CoV-2. All of these have the same kind of structure. And if you look at this kind of spherical-like molecule, and then what you can see in the rest of this is there's different proteins on the surface or on part of that capsid protein. And these are really important for different functions. One that's really important is called the spike protein. And this one I'll talk quite a bit about. This is the actual molecule that I mentioned previously that is important for this virus to infect the body itself. But there are other really important proteins. Some of these are found inside. These are nucleocapsid proteins. There are also proteins just right on the surface called membrane and envelope proteins, two different kinds. And then inside the core is where we find that RNA, that viral genome. So this is where the actual genetic information is kept. So what you should realize is that all of these, except for the viral genome itself, besides that RNA, all of these are actually potential antigens. So all of these are things that could develop an antibody against them. And that becomes really important in how we fight this disease, but also how we detect the disease and how our bodies respond to it. So just a little bit more about SARS-CoV-2. How does it bind and how does it enter and infect our body? So if you look at this structure, and I'm going to just blow up this little portion a little bit. So we have this spike protein. And you see it's actually three different colors are just kind of the three different domains or pieces of what make up the spike protein. And right in the middle of that spike protein is what's called the receptor binding domain. Or the RBD. So this part is really important for latching on to something in our body and then using that to shuttle itself into the cell. And so the particular protein that happens in our body, that it attacks in our body, is called ACE2. So that is the receptor that it binds. So inside our body, if we have this kind of an infection, the SARS-CoV-2, the spike protein binds to ACE2, and then, as I mentioned, it kind of hijacks that and gets inside of the body, and then eventually other parts help in viral reproduction. So what does that mean in terms of antibodies? Okay, so I mentioned that all of these different parts, the envelope, the membrane protein, and the spike protein, even the nucleocapsid proteins, all of these could be antigens. look at the mechanism for how this is actually infects our body, and then stop and think about, okay, if my body develops an antibody against, so if my immune system develops an antibody against COVID, what is the best protein for it to develop an antibody against? Well, do a little kind of just mental game. If you had an antibody that was bound to the membrane protein, how would the immune system see that? What would it do to that? Well, the membrane protein is not important in infection, but it is very important in structure. So if there was an antibody attached to this membrane protein, then potentially the body could see that and use it as some kind of opsonization mechanism or some kind of complement activation, and then kill this directly. And that does happen. But what I think is much potentially more effective is if you have an antibody that blocks the spike protein, more specifically, if it can block the receptor binding domain. If it binds right at that domain, then what happens? This RBD can no longer interact with ACE2. And so if RBD can't bind to ACE2, no infection. So this is what we actually refer to as a neutralizing antibody. And so if you hear in the news about these idea of neutralizing antibodies, that's what we're talking about. It also becomes really important when you stop to think about, antibodies in terms of therapeutics. So, a lot of people that have been infected are very have this desire to donate plasma, thinking that their antibodies will help somebody else. And this might be true, but you can imagine a person that has antibodies and has cleared the infection against the membrane protein might not be as effective as somebody who has neutralizing antibodies in their plasma. And so it's important for us to distinguish between those different types of antibodies. So not all antibodies are equal. Now, I did mention that because all of these are potential antigens, so all of these are what we call antigens. If we were looking for an assay, there's three different major flavors of assays for COVID-19 right now. And one of them is antigen-based assays. And so what antigen assays are looking for, these diagnostic assays, they are looking for one of these structures. It depends on which assay it actually is. The majority of them are designed to look for the spike protein, but there are assays that also look for membrane and other structures. And so they're looking for pieces of the actual virus in our blood. And so these are the antigen tests. The second type are PCR tests. And so these PCR-based tests, these are looking for are the actual RNA inside that's leaked out into our blood. And we should be able to, or in the virus itself, that can be disrupted, and we can pick that up. And if it's present, that means we have some kind of an infection. So a lot of those PCR-based tests, that's what that is looking for. The third type are actually antibody tests. So antibody tests are interesting because it will tell you if you have developed an immune response to COVID. Now, that doesn't happen right away. So if you just got infected with COVID, you probably won't have any kind of antibody yet. As your infection goes on over a few days, weeks, you'll start to develop those first runs, the IgMs, and then eventually they'll transform into IgG type tests. So a lot of the antibody tests that are out there, they're very useful for us identifying people that have had an infection, but might not still have that infection. And also the difference between what stage they're at. We can kind of determine what stage they might be at. So I want to talk about one specific antibody test that we've actually developed in my lab in collaboration with some excellent people at the University of Toronto. And essentially what this is looking for is what we wanted to do is develop an antibody test that could be very quick, that we didn't have to do any kind of long processing like many of the other antibody tests. And so what we developed is a system where we have what we call a split luciferase. And stay with me here for a second. What a luciferase is a protein that produces light. Many of us have experienced these in terms of firefly, where you see that light that's expressed. That's a protein that's doing that. And what we did is we engineered a specific luciferase to be in separate parts. And as long as these parts are separated, then there's no light that can be produced. But if we can force these pieces to come all together, then the light will turn on. And so what we did is we actually took part of this this luciferase and infused it to something that will bind to antibodies. And then we took another part of this luciferase and fused it to an antigen. In this case, what I'm talking about is an RBD. So if we take the RBD, the receptor binding domain of COVID, and fuse it to part of the luciferase, then we have something else that will kind of sandwich onto the antibodies. Then if we take somebody's plasma that has potentially a patient that has been exposed to, or one of us that have been exposed to COVID-19, if there are IgG in there, then this is what should happen. What should happen is the IgG itself will bind to the receptor binding domain. And then the other piece of our assay will bind to the antibody. And in the middle of this, it'll sandwich everything together and it'll force the luciferase to turn on. And so by doing this, we can actually see quite well, not only if there are antibodies present, but we can see how many antibodies are present. I know this is a very busy slide. I just want to walk through this very simple kind of steps. What we can do is we can take all of our components and mix them in with serum. So we have these different fragments that can be mixed together. These bind to the antibody and give us a signal that we can read and look for that luminescence. A lot of these are different kinds of controls that we wanted to see if it's specific to humans and if it's specific really to COVID antibodies. And so this is what we do. If we're doing science well, we really try to make sure that it's not only working, but it doesn't work when it shouldn't be. And so this is a different kinds of controls to make sure our assay is working correctly. But I'm most excited about this graph on the far right. Well, this is about 100 different samples from patients that we took, again, with the collaboration from Igor Segler's lab up at the University of Toronto. These are excellent people that we've been working with. And then my lab here at the University of Utah. And so what we have is taking patient samples at different times post-infection. And so this is how bright that light is, how bright the signal is for patients that are the serum samples before the pandemic. And then you can see about day 1 to 5 after infection, we don't have really too many signals. But what we can see is as time moves on, day 6 to 10, we start to have more people that have a signal. So what does that mean? Well, it means more people are starting to develop antibodies against this infection. And you see this really turns on at 1 to 2 weeks post-infection, stays quite robust at 16 to 20, and it persists for quite a long time. Some of these samples are 3 to 4 months after infection. Some of them do start to decrease a little bit, but the vast majority of people that have been infection maintain those antibodies for several weeks, several months after their initial infection. And we're able to quantify those quite quickly with this kind of an assay. But there's a lot of these kinds of assays out there. There's a lot of kind of assays that will tell you, yes, there's an antibody present. What we really wanted to do is take that one step further, and we wanted to say, can we detect if there are neutralizing antibodies specifically? And we want to see how good of neutralizing antibodies they are. What that means is, okay, an antibody that binds to the spike protein and blocks ACE2 from interacting, that's a neutralizing antibody, right? But some of them might bind really tightly, some of them might bind a little bit off the center of that RBD, and so they might not be as effective as neutralizing antibodies. So we want to say not only do we have neutralizing antibodies, but how potent are they in these individual samples? And so what we did is we redesigned our assay just a little bit. Now what we do is recombinantly fuse ACE2 with part of that split enzyme, and then we take the spike protein, or at least the receptor binding domain of the spike protein, and fuse it with the other part of the split enzyme. So now, if these pieces come together, everything will turn on. But if there's a neutralizing antibody present, we lose the light turns off. So however much inhibition, however much blocking of this interaction happens, that is directly proportional to the concentration and the potency of this antibody. And this is just a very early study using a control antibody. This is an antibody that's been shown, at least by the supplier. This is what they did to check to see if this is active. And so this is saying the higher concentration of the antibody, the less activity we get. And that's what we want. We want to shut down the activity. And so we did the same kind of an assay where we took these different controls, a couple different constructs, and showed that as they increase the concentration of the antibody, we can block activity of the ACE2. Now, that's only important if we can do it in patients. And so we did a pilot study with about 18 different patient samples. we were able to, with our assay platform, we can take direct from plasma. So we can take the plasma, we mix it in with our different reagents, and then we run to see what kind of output we get. These red lines right here, those are kind of the controls saying that we know that this is the level of signal we should get from this kind of a neutralizing antibody. Now, what's really intriguing is this is a whole group of different patients we didn't know anything about. All of them were in the ICU at some time with an infection. And so we tested all of these different patients. We did know, however, that patient number 16, 17, and 18, they all had antibodies. That's all we knew is that they had them. And we're really excited when they started to show up. Here you can see this patient number 17, and here is 18. And all of these, and then 16, a little bit, maybe not as strong, but what we can see is not only do they have antibodies, but they are neutralizing antibodies. These are the kinds of antibodies we really want to see. So moving forward with COVID, why do we want to keep track of neutralizing antibodies? People aren't getting infected anymore, right? Well, I think this is also a really excellent way to monitor vaccines. So it turns out, just because you've had, like I mentioned, just because you've had an infection doesn't mean you have maybe the best type of antibodies, or at least these neutralizing antibodies. But the vast majority of vaccines that are developed, they all are presenting the immune system with that receptor binding domain, the vast majority of the vaccines. And so that means that the kind of antibodies you'll develop if you take a vaccine should be the best kind. So to me, that really makes me think that, okay, even if I've been infected with COVID, it still makes sense for me to get a vaccine to ensure that not only do I have protection for recovering, but I have this added protection just in case I didn't have neutralizing antibodies. So the value of an assay like this in that kind of a context would be, we can now start to assess, these vaccines. How well are they producing neutralizing antibodies? How long do these antibodies last after they've been vaccinated? And using that, then we can help guide some of these clinical decisions. Do we need booster shots in the future, right? As variants come out, as the structure of the virus itself changes a little bit, do antibodies that were used in the past antibodies that we developed in the past still work against these new variants or not? And just by changing a few things in our assay, we should be able to kind of monitor those things. So there still is a value moving forward. The other thing I really wanted to mention before I move on to some of the therapeutic importance of antibodies is that here at the University of Utah, there is a lot of effort into researching COVID-19. And I really strongly encourage you to take a few minutes to browse the website for the III. We call this Immunology, Inflammation, and Infectious Disease Group. And that is really the consortium that has come together for bringing experts from all over campus to really kind of study this disease, and especially in the context of how it affects people locally as well. So take a few minutes to browse that if you're interested, and look at some other amazing research that's going on here at the University of Utah. So on to antibodies, and how can we make drugs out of antibodies? So how can we actually hijack this whole system for our own desire? Now, one thing that I'm going to show you is I like to call this a party trick. And what I want you to notice is the names of all of these different kinds of drugs. So these are the top selling drugs from 2019. These are based on billions of sales. So that is 19.7 billion with a B. And what you'll notice is these are all very expensive, but that also means they have a lot of sales. Focus in on the name, not this trade name, but what we call the generic name. And you'll start to see something coming up over and over, and that's MAB, M-A-B at the back of this name. So you see it here, adalimumab, pembrolizumab, rituximab. So MAB is an abbreviation for monoclonal antibody. It's called a monoclonal because it was produced to be one type of antibody and only one type of antibody. But what you can now tell your friends is next time a commercial comes up and you're watching the person riding their bike with a little plaque psoriasis that they're worried about, and they're being treated with Humira, and you see it pop up as adalimumab, you can turn to people in the room and say, that's an antibody, and just impress them to death. So what I think is also important is if you look at all of these top sellers, these are the ones that are antibodies or antibody derivatives. So the vast majority of the top selling drugs in the United States are antibodies. so these are an important class of drugs, but I want to dig into a little bit about what we know about them and how my lab is really trying to focus on making them a little bit better. So looking at this idea of antibody as therapeutics. So again, this idea of monoclonal antibodies, MABs, you might hear this MABs interchangeable with antibodies therapeutics as I move forward. So there's a few mechanisms we've already mentioned. So opsonization, neutralization, complement activation, this ADCC, antibody-dependent cellular cytotoxicity. These are the mechanisms we know about that our immune system already uses. So in essence, really what we could do is just create an antibody that's specific for one of these functions and hijack it that way. But there's also some non-standard mechanisms that we can start to put together and design new molecules that can do not only these things, but maybe something on top of the standard kind of mechanisms for antibodies. So starting with rheumatoid arthritis, this is one of the most prevalent diseases in the United States. So rheumatoid arthritis, Crohn's disease, ulcerative colitis, they all share one common element. What you can see here is in kind of a healthy joint, you'll see not very much of this thing that I show, but in the inflamed joint, there's a whole bunch more. What this is it's called TNF. The name is tumor necrosis factor, but it's a very, very important molecule in the immune system. So it's one of these other kind of signaling molecules that tells the body it needs to be doing something. In healthy joints, not very much. In disease joints, high levels of TNF. And this is true not just in arthritic joints, but also in other locations for Crohn's and ulcerative colitis. So this TNF that I mentioned, what does it do? Well, it does a ton of things. And I'm not going to go into each one of these mechanisms, but feel free to look at some of the references if you want to learn more. But what is important is as a signaling molecule, it tells the immune system to attack. And it says there's something going on here. Either there's a wound, there's an infection, there's something happening here that I need to recruit other molecules, other cells to this environment to kind of try and fix what's happening. Now, unfortunately, when we have an autoimmune disease, these are diseases where we're attacking ourselves. And so something like rheumatoid arthritis, TNF is actually the instigator that's launching an attack and causing breakdown in our joints. So antibodies have come on the scene as a very potent therapeutic. Two of the most popular and well-known are adalimumab. You see, there's that mab again, which trade name is Humira, and infliximab, which trade name is Remicade. And so these are both IgG molecules, and what they do is they bind to TNF directly. And so if you can imagine, if it binds directly to TNF, then it blocks TNF. And so if TNF is kind of stuck onto an antibody, well, now it's neutralized. It can't interact with all of these other systems. It can't interact with the receptors that are on cell surfaces. So it can't pass its signal on anymore. And so what that should do is it should start to decrease the inflammation a little bit. It should pull back on some of these disease states. The problem with TNF is it is such an important regulator of the immune response that if we have too much TNF in an environment, then we have these autoimmune disease. But if we block it completely, that just opens the door for infections to happen. So it's really important that we have a balance somewhere in the middle. We don't want too much we don't want to block too much TNF, but we need to block enough TNF. And that becomes really challenging when we think about the body versus a specific location in the body. So you can imagine, I really want to block TNF in this environment at that inflamed joint, but I don't need to block it all the rest of the places in my body. So this is where the idea of drug delivery or targeted drug delivery really comes into play. What we want is to localize the therapy only where the disease is. So what was recognized by Michael Yu here at the University of Utah in the biomedical engineering department is that a lot of these sites of inflammation, they have other things besides TNF. So yes, if we look at an inflamed site, there's increased TNF levels, but there's also increased levels of damaged collagen. And so collagen is something that we might hear on the news. It's really a structural protein that helps keep everything together. So in healthy tissue, there's low levels of TNF and there's healthy collagen. So now we have something different between these two locations. We have high levels of TNF and we have damaged collagen. And so what Michael Yu and his team came up with was this idea of taking a collagen hybridizing peptide. So this is a small peptide that kind of mimics this collagen that's damaged. And what happens is if you take this collagen hybridizing peptide and you inject it, it's going to go and get stuck wherever there's damaged collagen, but it doesn't really get entwined with where the healthy collagen is. So that means using this, we should localize only in these disease sites. And you can see from this kind of a model, I encourage you to go read the paper where we did some of this modeling. It really shows up in some of these joints and other locations where there's a lot of remodeling going on. And so my lab, where we come into play, is this idea that, okay, well, putting the collagen, localizing this is only half the battle. And what we really want to do is take and say, we want the TNF itself to be blocked. So what we did is we took infliximab and we did a digestion. So we only want this part of the infliximab. We only want the part that is signaling the actual, or that is actually blocking the TNF. We don't need the rest of the molecule. So if we digest this, then we have just this antibody fragment, and then we can chemically conjugate or link these two pieces together to create a new hybrid molecule. And so with these hybrid molecules, what they are is this arm is going to block the TNF, but this arm really gets localized wherever the disease state is. So what hopefully we can do then is we don't have TNF blocking in other places where we still might need it, but only where it is actually overregulated and inflamed in the disease sites. So looking at an arthritic mouse, so we did some experiments where we actually took an arthritic mouse and we injected these different constructs into that mouse. And then we looked at their joints. So what I'll really highlight here is, looking at that joint, that's what we're showing in this picture. But right here at this interface, what the blue is where collagen is missing. So this red in the picture is collagen. This is where good collagen should be, that it's supposed to be intact. So if we treat a mouse with just a saline solution, the collagen doesn't get protected. It gets broken down and destroyed because of the TNF. But if we can actually take and get our stuff intertwined into this location, then over time, we start to see a rebuilding of this collagen deposition. So we're starting to repair what's been broken. And eventually that repair is really what will help stop the inflammation that is problematic. So we're continuing this work. We're trying different avenues, trying to get this system to work a little bit better, but hopefully can push forward to different new types of therapeutics that don't have some of those bad toxic or side effects that Humira and infliximab normally do. Another major area for antibodies are therapeutics. And so these therapeutics are in cancer. And so some of the main areas that we've seen emerge very early on were targeting things like HER2. So an antigen like HER2, where you can see this molecule on a cell surface. And HER2 is really important because it partners with other molecules and it signals things for the cell to do, including divide and survive and not die, which is exactly the hallmarks of cancer, these cells that just continue to grow. And so the idea is especially in breast and ovarian and gastric tumors, if we can block HER2, if we can bind an antibody here that maybe stops it from interacting with a partner or stops it from having this kind of an effect, or allows other macrophages or other immune cells to come and target the tumor and kill them directly, this would be excellent. And so there's many products out there, but two of them that specifically target HER2. One is called trastuzumab, Herceptin. The other is pertuzumab or Perjuda. They have a little bit different function, but both of them do target this HER2 molecule. Now, unfortunately, the antibodies by themselves don't have a real good potency. Again, what they do is kind of block an interaction, or they might signal some other cell type to come in and attack the tumor, but it's not extremely potent on its own. It really needs some kind of other molecule along with it. I want to talk about this a little bit more. And so I'm going to take just a second to look at a few of the different mechanisms for antibodies, specifically in cancer. So one that I just mentioned is this idea that antibodies can bind to a tumor and then they'll signal some other cell to come in and attack it. Another one is this idea that it blocks some kind of a signal that helps the tumors grow. In this case, there's molecules like VEGF that help the tumors grow. And if we can block that or kind of mop it up, then the tumors can't grow quite as quickly. The last one, so second one, third one is this checkpoint inhibitor. We hear a lot about these. So what this is doing is taking the brakes off the immune system and allowing them to go ahead and target the cancer. And again, if you want to learn more about these things, I really encourage you to go read these some of these reviews. They're very readable and provide a lot of information. Others that actually take antibodies, and this gets really interesting to me, where they take an antibody and then conjugate some other molecule to it. So maybe this helps with radiotherapy. Or other ones that are emerging like CAR T cells, or making hybrids of antibodies in cells now. There are also antibody drug conjugates, and this is really where our lab focuses a lot, and I'll talk a lot more about that in a second. But another kind is this bispecific antibody. So an antibody that has two different arms, and each arm reaches out and grabs something. One arm grabs the tumor, one arm grabs the cells in the immune system and brings those together so they can interact and combat the tumor. But these antibody drug conjugates, these are fascinating molecules to me, and really one of the reasons that I'm in academia. So what an antibody drug conjugate is, this is abbreviated as an ADC. So this takes this really large molecule, these antibodies, and then we stick small potent drugs on them. And so through these different linkers, we can link a drug to the antibody backbone. So why would we want to do that? Well, if you or anybody you've known has ever had to take some kind of chemotherapeutic, you'll know about all the horrible side effects. And that's because these drugs, they're active, and they're just distributing everywhere throughout the body. But what do we know about antibodies? We know that antibodies are really, really good at binding something specific. So what we can do is take these drugs, and now they're linked to the antibody, and they're only going to go where the antibody goes, which means they're not going to just disseminate throughout the body, but they're only going to lock on to those targets that we want them to. And so hopefully can really decrease some of those side effects and toxicities, but still have the potency of a small molecule drug. So these kind of ADCs, they should accumulate at tumor sites. They hopefully can protect normal tissue from some of the really toxic effects. And a lot of these ADCs are actually already built on some of the other molecules that I mentioned. So people can take trastuzumab, for example, like we have done, and conjugate a drug to it to make a new form of therapeutic. So I'm not going to talk about this on the left a lot, but I think it's important to mention that one thing that our lab does a lot of is characterization. So we not only make all of these molecules that I'm talking about, but we put a lot of time and effort into making sure that what we made is real. And that's all that I would really say about these different slides. If you want to know more about these, scientists, we're by nature pretty narcissistic. I would be happy to talk to you more about my work anytime, and just reach out to me, and I'll tell you about how we characterize these antibody drug conjugates. But one thing that we wanted to ask this question is, what happens if we take an antibody drug conjugate and combine it with a second drug? So a lot of these ideas that we're not going to just use ADCs on their own, but if we combine it with a second molecule can we get these additive effects? Can we get even synergy to happen between these two molecules? And so one project in the lab is to create these antibody drug conjugates that have these really elegant pieces, combining linkers that can be degraded and release the drugs after they've been taken into cancer cells, and combining it with some other small molecules. And what this graph is showing on the right, the bottom right, is how potent a molecule is. So this is how many cells it kills. So this is how many cells are still alive. So at low concentrations of drug, 100% of the cells still are alive. But as we increase the concentration, we add more and more antibody drug conjugate, we start to kill those. Now, what you'll notice is there's different shapes of these graphs. What's that's important is as we combined the antibody drug conjugate with the second drug, we start to see that we can actually push this a little bit farther down. We're able to kill more of the cells, and we're able to kill more of the cells with less drug. So by combining drugs, we're actually able to back off a little bit the dose that we needed originally, which again can help spare some of those side effects. Now, this particular combination turns out that if you go too high with some concentrations of small molecules, it starts to take over, and those toxic problems show up again. That toxicity that we've talked about with just having a small molecule. So what we decided to do is actually create two antibody drug conjugates. This is one approach we're exploring right now. So if we combine one antibody drug conjugate with one drug, with a second antibody drug conjugate, and both of these can simultaneously bind to a given target, then what we have is a system where we can overcome some of these off-target toxicities, but still have the potency that we want. And that's what we're showing in this graph. So moving on to kind of this last area of research in our lab, we already mentioned this idea of infection. And so the body is already really, really well equipped to use antibodies to fight bacteria and virus infections. So I would have thought that, and it turns out that if you want to go even deeper, there are a ton of different kinds of mechanisms that are available that can also be used to combat infection using antibodies. So I would have thought when I got into this that there would be a ton of antibodies out there to treat bacteria, but there are none. There are no FDA-approved antibodies to treat bacterial infections. There are a few antibodies that are out there that they can use to treat the toxins that these bacteria release. So things like cholera or anthrax. There's some antibodies that can block the toxin these things release, but they don't work very well against the bacteria themselves. And we're really curious about that. And we thought, at a minimum, what we should be able to do is take an antibody that we design against a bacteria, and we should be able to induce opsonization, and cells of the immune system should eat that, right? And so what we did is we tried that. So I'll just walk through this graph very quickly. So these are cells of the immune system, neutrophils that are not active. They're just kind of in hibernation mode. And you can see when we mix in the green, the bacteria that we've labeled, you don't have a lot of bacteria that are engulfed by the neutrophils. But if you stimulate the macrophages to turn them on, then they start to become more active and they're starting to eat a lot more of the bacteria that are there. However, if you take an antibody and add them in as well, then what you can have is a whole process where this just goes so much greater and really induces more of the opsonization. So we proved the point that yes, these do increase opsonization, increase the amount of cell killing we have of aeruginosa. This is aeruginosa, Pseudomonas aeruginosa. But I want to point out something. This is the amount of viable bacteria that we have remaining. And even in this approach, you'll notice there's still 25, 30% of the bug that's still alive. And so maybe this is one of the reasons that antibodies aren't used, is right, because these really resistant bacteria like MRSA and aeruginosa, they've developed mechanisms to escape this kind of process. So even though you get inside of a macrophage, you can't kill it. So what we're doing is we're combining this idea of antibodies, and again with antibody drug conjugates, but here we use antibiotics. So now what we're going to do is a kind of a 1-2 punch. In some cases, a 1-2-3 punch with 3 different types of drugs. So we create antibodies that have these different kind of antibiotics, and so then these first force the incorporation, the uptake by cells of the immune system. And once they're in there, even though they might be able to escape this first step, and they've developed mechanisms to resist digestion by the macrophages and neutrophils, what we have are now we've just released a whole payload of drugs right next to them that we predict they aren't going to be able to resist. And so we're really working hard right now to develop these kind of antibiotic conjugates to combat different things. So this is where I'll end. I think that what I wanted to really convey is that these antibodies are really versatile platform. There's tons of them. There's over almost 3,000 different mAbs that are in development right now. IgG is popular. There's a lot against cancer, but it seems like the highest selling ones are against TNF alpha. I do want to leave with, this is just the beginning for antibodies. We have this basic structure of an antibody, but what you can see is we can start to take pieces of the antibody and create new entities. We can actually combine them in different ways to create these new and elegant, brand new macromolecules that can have novel functions that might help bypass some of this resistance. So I'm really excited about antibodies, obviously, I've given you kind of a nice overview of our lab and the things that we do, how we use these platforms for different kind of systems. But I'd be remiss if I didn't stop and really point out the people that did a lot of this work. People like Andy and Sunjin that really work on the diagnostic side of things. Jessica that works on the antibody drug conjugates, and Christine that works on the antibody drug conjugates and antibody antibiotic conjugates. Keith that has done the work on the CHP, so the rheumatoid arthritis projects. Some very talented undergrads that we've had in the lab that have moved on or are still currently here. And I would like to thank our funding sources. And so with that, I really will end my presentation, and I'd really be happy to take any questions that people might have.
Awesome. Thank you, Dr. Owen. And I will kind of jump back on and help with the questions. It was fascinating to hear lots of the cool things that your lab is doing. And it makes me really excited to be entering pharmacy right now and being a patient in the future, because it gives me so much optimism. So thank you. And thanks for all the work that you're doing. So a lot of questions that have come in so far are surrounding COVID. So we're going to jump back to that if you're all right.
Let's do it.
Awesome. So here's our first one. With the current information, how long is the virus vaccine going to last? And will we need boosters at some point?
I am not an immunologist and not a virologist, so I'm going to half dodge that question. What I say is that from our personal experience, we have seen people that have been infected continue to have antibodies months after their initial infection. That gives hope that there's kind of this memory that's in there. So even if another infection comes, if the virus hasn't changed too much, there should be some level of protection. As far as vaccines go, we don't have that much information, but there's some really promising results that look at this idea of persistence, where antibody memory seems to be there. And so I think my personal prediction is eventually that might start to fade, and with new variants that come online, we might have to have a booster eventually, but I think it's going to look a lot like our flu vaccine now, that each year we go and we get that additional booster shot, or even it might even end up something like a tetanus booster shot, where every 2 to 5 years we need to go in and get it re-jumpstart the immune system. But it's hard to say for sure right now.
That's cool, and definitely optimistic and exciting to see the vaccine roll out more and more. And hopefully meaning a return to normal for us. Okay, question 2. Do all viruses have your body create antibodies?
Theoretically, yes. The problem is time. And severity. So unfortunately, what happens, sometimes your body doesn't make it in time. And so this is why it's really important. This is the idea of a vaccine. The different types of vaccines that are out there is we're giving parts of either parts of the actual virus, so we can show it to the immune system and say this is what to expect, but it's not actually infecting and causing all those other problems. So the virus overburdens the body, and it just doesn't have time to make an antibody. If given enough time, if given just parts of the virus instead of the whole thing, maybe it should be able to recuperate and do that system.
Very interesting. Thank you. Here's our third question. Do things like severity of illness or whether someone's symptomatic or asymptomatic affect antibodies?
This is a great question. And this is one that I ask myself. And because we really have been limited in differentiating a couple of things, how much antibody do we have and which type of antibodies do we have? Most of the tests out there right now are yes, no. It says this person has antibodies. That's it. But I think in order to answer this kind of question, these asymptomatic carriers, why are they asymptomatic? Does it have something to do with the antibodies or not? I don't know. But if we can actually, start to pull out and say this person was asymptomatic and they had this type of antibody, then maybe we can start to make a correlation like that. So it's a question we're looking at as well right now. So great question.
Oh, that's cool. And it's cool how relevant all of this is in your work and the, pandemic and everything like that. So thanks. So this one came in during our meeting. So it talks about an antibody test, and you can answer it for your test that you're working on or other ones that you know of. But will the antibody test differentiate antibodies from a COVID-19 infection versus the antibodies from an mRNA vaccine?
That is an interesting question. It really depends, is the answer. So most of the way that antibody tests work is that most of the tests, they have to have something for the antibody to lock onto. And so essentially what you're doing is you're taking a small piece of the virus that we've made in a lab and then looking to see if anything sticks to it. And so if, let's say that we have a person that was infected versus a person with a vaccine, and they both have an antibody that binds the same part of that, I wouldn't be able to distinguish whether that was a naturally arising one or from an infection or from other system. What I would be able to tell is that because these, like the mRNA vaccines specifically, we know exactly what part of the protein it is. So it's very easiest for us to predict what types of antibodies we see. So if we did an assay and saw those, and then also saw some other antibodies that were against maybe a different part of the protein or some other thing, then I would conclude this person had an infection that gave them one type, and the mRNA gave them a different type of antibody. That's why personally I'm encouraging even people that have been infected, get the vaccine anyway. It's double protection. It's making sure that you have the most effective types of antibodies that might be out there.
Very cool. Thank you. This question just came in, and I think it's a good one. How are antibodies taken into the body without being broken down in the gut?
They aren't. So there are currently no oral formulations of antibodies. This is kind of the holy grail. There's some really interesting things coming online, even little micro robots that are trying to inject antibodies at a specific place in the gut, but you can't take an antibody orally. It's a large protein. It will be broken apart. There's a very nice work of using antibody surrogates, things that are kind of like antibodies, but might not be broken down. By people like Michael Kay in the biochemistry department here at the University of Utah, that can function like an antibody and can be taken orally. So kind of mixing both worlds. All of the antibodies I talked about, all of those therapeutic approaches are all going to be subcutaneous injections or IV injections at this point. I think it's a very interesting field to push forward though.
Yeah, no, super interesting and super relevant. Thanks. All righty. So we will go ahead and wrap this up. We would like to thank especially Dr. Shawn Owen for his presentation today. I know I learned a lot, even as a current pharmacy student. So super thankful for him, for all the research that he's doing and the things he's doing to propel medicine forward. So thank you to Dr. Owen and to everybody in his lab and at the University at the College of Pharmacy. And thank you everybody for joining us today. The Alumni Association and the Salt Lake Alumni Club does host weekly events. So this is a great chance to take a lunch break, go ahead and eat while you're watching, and enjoy learning a few things and engaging with the Alumni Association here at the U. So thanks for everyone for joining us, and have a great afternoon. Go Utah and go Utes.
Okay, we are all done.